Welcome back everyone. Today we have going to discuss how gametogenesis occur, right? We know that the male gamete is the sperm and the female gamete is the ovum, right? And we have seen how the male reproductive system is made of and how the female reproductive system is composed of, right? We have seen how the female and male reproductive system is. Now we are going to see how gametogenesis occur, right? How gametogenesis occur, right? So the first thing we need to know that we have, which is a male gamete. Sperm is the male gamete, right? And which is a female gamete? It is the ovum, right? We all know. Sperm is the male gamete and ovum is the female gamete, right? Where the sperm is produced? The sperm is produced in the testis, right? Sperm is produced in the testis, right? In male, sperm is produced in the testis. And what about ovum? Ovum is produced in the ovary, right? And the process by which the sperm is produced in the testis is called as what? It is called as spermatogenesis, right? It is called as spermatogenesis, right? Right? So in gametogenesis, right, we have now which is a male gamete. Male gamete is the sperm and the female gamete is ovum, right? So the male gametes are produced where? It is produced in the testis, right? So the process by which the sperms are produced in the testis is called as what? It is called as spermatogenesis. And the process by which the ovum is produced in the ovary is called as what? It is called as oogenesis. Okay, oogenesis. Okay, so in humans we need to see how spermatogenesis occur and how oogenesis occur. Okay, how spermatogenesis occur and how oogenesis occur. Okay, so before going into spermatogenesis and oogenesis, I want all of you to know an important point. What is that? The sperms or ovum, whatever the gamete is, it is produced from a single cell. What is that cell? It is the primordial germ cell. Okay, primordial germ cell. Okay, so the sperm as well as the ovum is produced from which cell? These are produced from the primordial germ cell. Okay, the primordial germ cell, right? This primordial germ cell, where can we see the primordial germ cell? We can see the primordial germ cell in the embryo, right? The primordial germ cell are present in the embryo or present in the embryonic development stage. Okay, the primordial germ cells are present at the embryonic development stage. So what happens to this primordial germ cell? It will migrate. Okay, it will migrate to the respective gonads. Okay, in males it will if the baby is a male baby, right, the primordial germ cell will migrate to testis, right? It will migrate to testis, right, and give rise to sperms, right? In the embryonic development, if the baby is a male baby, right, what is meant by male baby? It has which chromosomes? It has X and Y chromosome. Okay, it has X and Y chromosomes. What happens? This primordial germ cells will migrate to the respective gonads, right? In male baby, it will migrate to the testis and form sperms. If it is a female baby, right? What if it is a female baby which has XX chromosome? What? The primordial germ cell will migrate to where? It will migrate to ovary, right? And give rise to ovum. Okay, so where the sperms are produced in this testis? It is in the seminiferous tubules. Right, it is produced in the seminiferous tubules. And in the ovary where? It is in the ovarian follicle. Okay, it is in the ovarian follicle. So the primordial germ cell which is present at the embryonic stage, what happens? According to the sex of the baby, if it is XX chromosome or XY chromosome, the primordial germ cell will migrate to the respective gonads, right? If it is a male baby, it will migrate to the testis into the seminiferous tubules to give rise to sperms. If it is a female baby, it will migrate to the primordial germ cell will migrate to the ovary, to the ovarian follicles to give rise to the ovum. Okay, this process of the formation of sperms in testis is called as what? It is called as spermatogenesis, and the formation of ovum in ovary is called as oogenesis. Okay, we will see one by one. Okay, we will see one by one. Okay. First, which is the first one? We have the spermatogenesis. Okay, Spermatogenesis, right? When I discuss the male reproductive system, right? When I discuss that, I told you what? I told you about the okay. I told you that the seminiferous tubules, right? The seminiferous tubules is lined by male germ cell and Sertoli cells. Do you remember? Right, I told you that it is lined by male germ cells and Sertoli cells, right? Male germ cell, right? And this is the Sertoli cells, right? Male germs, seminiferous tube. This is the seminiferous tubules. Okay, so the male germ cell is called as what? It is called as spermatogonia. Right? From which cell these spermatogonias are formed? The spermatogonias or the male germ cells are formed from the primordial germ cell, right? Primordial germ cell. So what happens at the time of puberty? Okay, when the baby is delivered and it has grown and reaches the puberty, what happens at the time of puberty? This spermatogonias, okay, we have this spermatogonia. This spermatogonia will starts to undergo what? It will undergo mitotic division. Okay, I told you the seminiferous tubules is composed of Sertoli cells and the male germ cell. This male germ cell is called as what? It is called as the spermatogonia, right? So at puberty, this spermatogonia, okay, they will undergo what? They will undergo mitotic divisions. Not divisions, okay? If a number of cycles of division, okay? The spermatogonia will undergo mitotic division, okay? And they will form what? They will form type A spermatogonia and type A spermatogonia daughter cells and type B spermatogonia daughter cells. Right? Type A spermatogonia daughter cells and type B spermatogonia daughter cell. So what will be the chromosome number in spermatogonia? Remember that the chromosome number in spermatogonia is 46 or 2n, right? 2 pairs, diploid. Spermatogonia is a diploid cell, okay? It is a diploid cell. And what is the chromosome number? It is 46, okay? 2n. Spermatogonia is a diploid cell. It has 46 number of chromosomes, okay? So what happens when it undergo mitotic division? There is no change in the chromosome number, right? The type A spermatogonia and type B spermatogonia, what will be their chromosome number? It is 2n only, 46 only, because it's undergo mitotic division, right? And this type B spermatogonia, they will start increase in their size, okay? They will start increase in their size and forms what? Forms primary spermatocyte. They will form what? They will form primary spermatocyte, okay? They will form primary spermatocyte. So what will be the chromosome number in primary spermatocyte? It is only growth, right? The type B spermatogonia increase in their cell size, right? And form what? Form primary spermatocyte. So what will be the number? It is also 2n, right? Diploid cell, 46 number of chromosomes. Understand? Okay. Now this primary spermatocyte, okay, this primary spermatocyte will undergo what? It will undergo the first meiotic division, meiosis 1. Okay, it will undergo the first meiotic division, okay, meiosis 1, and forms what? And forms 2 secondary spermatocyte, okay. secondary spermatocyte. Right? 2 secondary spermatocyte. Which division? Meiosis 1. Okay. So what happens in meiosis 1? There is reduction in chromosome number, right? There is reduction in chromosome number. So what will be the chromosome number? Secondary spermatocytes are haploid, right? It has undergo first meiosis. In first meiotic division, what happens? There will be reduction in chromosome number. So what is the chromosome number in secondary spermatocyte? It is 23. Okay, 23. Okay, this secondary spermatocyte will undergo what? They will undergo second meiotic division. Okay, they will undergo meiosis 2. Okay, meiosis 2. And give rise to what? And give rise to spermatids. Here also 2 spermatids. What is the chromosome number in spermatids? It is haploid. Okay, spermatids are haploid, right? They are haploid. Okay, this is called, that spermatids is formed. That process is called as what? The process of formation of spermatid from the spermatogonia is called as what? It is called as the spermatogenesis, right? So from the secondary spermatocyte, right, the secondary spermatocyte will further undergo the meiosis 2, right? We have seen that there are 2 portions in meiosis, right? Meiosis 1 and meiosis 2. In meiosis 1, what is happening? There will be reduction in the chromosome number, right? There is reduction in the chromosome number. So the primary spermatocyte will undergo meiosis 1 and forms what? And forms secondary spermatocyte, right? So what is the chromosome number in secondary spermatocyte? It is haploid, 23 number of chromosome, right? The secondary spermatocyte will undergo the second meiotic division to form spermatids, right? Spermatids. And what is the chromosome number in spermatids? It is haploid, right? 23 chromosomes are there in spermatids, right? So each secondary spermatocyte will give rise to 2 spermatids. Okay, now the question arises. Okay, now we have the question, right? Now I am going to ask the questions to you, right? So how many spermatids are or how many sperms? The spermatids will further develop into sperm. So how many sperms will be produced from a primary spermatocyte, right? So from a primary spermatocyte, how many sperms are going to produce? We will get 1, 2, 3, 4, right? We'll get 4 sperms, right? We'll get 4 sperms from a primary spermatocyte, right? We'll get 4 sperms from a primary spermatocyte. What about from a secondary spermatocyte? Secondary spermatocyte, we'll get 2 sperms, right? Because 2 secondary spermatocytes are formed from 1 primary spermatocyte. Each secondary spermatocyte will give rise to 2 sperms. Okay, so the secondary spermatocyte will give rise to 2 sperms. Then the next question, how many sperms are produced from a spermatogonia? How many sperms are produced from a spermatogonia? Can you tell me the number? We can't tell the number because what? The spermatogonia will undergo a number of mitotic division. Okay, it is not a single mitotic division. It will undergo n numbers of mitotic division. Okay, a few numbers of mitotic division, right? Will undergo few numbers of mitotic division, right? So what happens in mitotic division? There will be increase in number of cells, right? Increase in number of cells. So there will be increased in the type A and type B spermatogonia. We don't know that number, right? We don't know how much division has this spermatogonia has undergone, right? So we can't tell the number of sperms from a single spermatogonia. We can't tell the number of sperms produced from a single spermatogonia. It will be anyway, it will be more than 4, right? Because the spermatogonia will undergo a number of mitotic division to increase in number of spermatogonias, right? So we can't tell how many spermatogonia will give rise to or how many primary spermatocyte is formed from spermatogonia? We can't tell that, right? Because the spermatogonia will undergo mitotic division to produce type A and type B spermatogonia, right? Out of which this type B, okay, the type B spermatogonia will form what? Will form the primary spermatocyte. Okay, so we'll just go through the steps once again. What happens? The seminiferous tubules has what? The seminiferous tubules has male germ cell or the spermatogonia and Sertoli cells, right? This we all know from the last video, right? It has the male germ cell and Sertoli cells. So this male germ cell is called as what? It is called as the spermatogonia, right? So at puberty what happens? At puberty the spermatogonia will start undergoing mitotic division and forms what? And forms primary type A spermatogonia and type B spermatogonia. This type B spermatogonia starts to increase in size to form what? To form primary spermatocyte. Till here the number of chromosome is 2n or diploid or 46 number of chromosome. The primary spermatocyte will undergo the first meiotic division. Okay, meiosis 1. There will be reduction in number of chromosome to form what? To form the secondary spermatocyte. So what is the number of chromosomes in secondary spermatocyte? It is haploid N or 23 chromosome. Okay, and this secondary spermatocyte will complete the second meiotic division to form spermatids. Okay, from this spermatid sperms are formed. Okay, from this spermatid sperms are formed. So how many sperms are formed from primary spermatocyte? 4 sperms or 4 spermatids are formed from the primary spermatocyte. How many sperms are formed from the secondary spermatocyte? 2 sperms or 2 spermatids are formed from the secondary spermatocyte, right? Spermatogonia we can't tell, right? Because it undergo a number of mitotic divisions. So we can't tell the number of sperms or spermatids or spermatozoa. Okay, understand what is spermatogenesis? Formation of spermatozoa from spermatogonia is called as what? It is called as spermatogenesis. Okay, so what we got? We have got the spermatids, right? We have got the spermatids, right? These are oval or round cells, okay? The spermatids are, right? These are oval or round cells, right? Oval or round cells with haploid number of chromosomes, that is 23 number of chromosomes, right? From this spermatids what we need? We need a sperm, right? We need a sperm, right? This is the sperm. We need a sperm, right? From this oval spermatids we need what? We need a sperm, right? This is the sperm. Okay. And the process of formation of sperms or spermatozoa from the spermatids, it is called as what? It is called as spermiogenesis. Okay, spermiogenesis. Okay, the process of formation of the sperm or spermatozoa from the spermatid is called as what? It is called as spermiogenesis. This is called as spermiogenesis. Okay, so we'll see what is happening. Okay, we'll see what is happening to the spermatids, right? In spermatids we have a central nucleus, right, which is haploid in number. Okay, and we have cytoplasm, right, surrounded by the plasma membrane, right? There will be cell organelles will be present, right? What are the cell organelles will be present? We will have Golgi apparatus, we will have mitochondria, right? We will have lysosomes, we will have all other cell, right? Centrioles, all cells will be, or all organelles will be there inside the spermatids, right? Say normal animal cell. What happens now? Now what happens is that the nucleus, okay, first what happened is that the Golgi apparatus will starts to produce increased number of lytic enzymes. Okay, there will be increased production of lytic enzymes. Okay, increased production of lytic enzymes. Okay, increased production of lytic enzymes. Okay, and there will be morphological changes in the shape of the cell also. Okay, there will be morphological changes in the shape of the cell also. The oval or the round shaped cell starts to elongate. Okay, they will starts to elongate, right? The cell will starts to elongate, right? And the lytic enzymes which is produced by the Golgi apparatus will starts to accumulate in one end of the cells. Okay, they will starts to accumulate in the one end of the cell, right? Then what happens? The nucleus, right? The nucleus present in the cell will starts to condense. Okay, the nucleus present in the cell will starts to condense and such that the nuclear materials become tightly packed. Okay, they become tightly packed and the central nucleus will be get shifted to one side. Okay, the central nucleus will get shifted to one side, right? The nuclear material become tightly packed so that the size of the nucleus reduces. Okay, they become condensed and tightly packed and the nucleus gets shifted to one side of the cell. Okay, then what happens? Then the mitochondria, okay, the mitochondria starts to arrange. Okay, they will get arranged in the, so the mitochondria will starts to get arranged in the cell. Okay, in the cell around an axoneme. Okay, around an axoneme. Okay, around an axoneme or axial apparatus of the cell. From where this axial apparatus is produced? This axial apparatus is produced from the centriole of the cell. Okay, the axial apparatus, this is the centriole. Okay, so from this centriole an axial apparatus is produced and the mitochondria, okay, the mitochondria get arranged on either side of the centriole of the cell. Okay, centriole of the cell, understand? Right. The Golgi apparatus, right? The Golgi apparatus, they will form a cap. Okay, the Golgi apparatus will be form a cap at the top end or at the upper end of the cell. Okay, the Golgi apparatus will form a cap at the upper end of the cell. Okay, upper end of the cell, which contains what? Which contains the lytic enzymes. Okay, the Golgi apparatus will form the cap, okay, which contains the lytic enzymes. Okay, and there is the nucleus, right? There is the nucleus which is condensed and formed at the one end of the cell, right? Which is formed at the one end of the cell. Then we have the mitochondria which is arranged around the axial apparatus or axoneme of the cell, which is formed from the centriole, right? And this centriole, okay, the microtubule, okay, there will be elongation of a microtubule from the centriole. Okay, there will be elongation of the microtubule from the centriole, right? It will forms the tail. Okay, so the nucleus part will form the head. Okay, the part which has the nucleus will form the head. The part with the Golgi body, okay, the Golgi body will form the acrosomal cap. Okay, it will form the acrosomal cap, right? Then the part which has the nucleus will form the head, right? The part with the mitochondria will form the neck, and the microtubule extending from the centriole will forms the tail. This is the structure of a sperm or a spermatozoa. This is the structure of a sperm or spermatozoa. So see, from this cell, from an oval cell, and what happens to the extra cytoplasm and other cell organelles? They are discarded. Okay, they are discarded. The extra cytoplasm and the cell organelles are discarded, or they get phagocyte by which cells? Which cells are there in the seminiferous tubules? Our Sertoli cells, right? So the extra cytoplasm and the cell organelles are discarded or phagocyte by the Sertoli cells. Okay, so this cells, which is a round oval shaped cell with a central nucleus, what happens? The Golgi apparatus will starts to produce the lytic enzymes, and the Golgi apparatus will form an acrosomal cap with the lytic enzyme at the one end, right? The nucleus will get condensed. The nuclear material will be tightly packed. Okay, the nucleus will get condensed and the nuclear material will be tightly packed and forms the head of the sperms. And the mitochondria get arranged on either side of the axoneme. okay, which is formed from, which is an elongation formed from the centriole, okay, axonium, and that forms the neck part. And what happened? The elongation of the microtubule of the centriole will forms the tail, okay, will forms the tail, right? So the spermatids will be get transformed to a spermatozoa and sperm or sperm. This process called as what? This process called as spermiogenesis. Okay, this process called as what? Spermiogenesis. So I told you that this spermatogenesis is occurring, or spermiogenesis, where these are occurring? These are occurring inside the seminiferous tubules, right? These are formed inside the seminiferous tubules, where we have the Sertoli cells, we have the germ cells. Okay, so what happens? So what happens? The sperms, okay, the sperms which are produced, okay, the sperms which are produced in the seminiferous tubules will have their head, okay, will have their head embedded inside this Sertoli cells, okay, they will have their head embedded inside the Sertoli cells with their tail towards the lumen of the seminiferous tubules. Okay, the head will be embedded inside the Sertoli cells, right? Because where will the sperms get nourishment? Get nourishment from the Sertoli cells. So the head will be embedded inside the Sertoli cells, and the tail will be present in the lumen of the seminiferous tubules. Okay, and the release of this head from the, okay, the release of this head from this Sertoli cell. Okay, the release of the head of the sperm from the Sertoli cell is called as what? It is called as spermiation. Okay, the release of head, okay, the release of head of sperm, or release of the sperms from the Sertoli cell, right? From Sertoli cells, it is called as what? The release of the sperms from the Sertoli cell to the lumen of the semin, or to the accessory duct, is called as what? It is called as spermiation. Spermiation. Okay, so after spermatogenesis we have seen two processes, right? We have seen two processes. One is what? One is the spermiogenesis. What is meant by spermiogenesis? The formation of a sperm or spermatozoa from a spermatid is called as what? It is called as spermiogenesis. There is morphological changes that occur in the spermatids to form the sperm. That is called as what? That is called as spermiogenesis, right? So the sperms or spermatozoa formed, okay, the formed are embedded inside the Sertoli cells, right? They are embedded inside the Sertoli cells, right? So the release of the sperms from the seminiferous, from the Sertoli cells, okay, from the Sertoli cells is called as what? It is called as spermia. So the process of conversion of the spermatid to spermatozoa or sperm is called as spermiogenesis, and the release of the sperms from the Sertoli cells to the accessory ducts is called as spermiation. Understand? That is called as spermiation. Okay, now we will see how the spermatogenesis is regulated. Before that I told you what? The spermiation, right? The sperms are released, okay? The sperms are released, spermiation occurs, right? The sperms are released from where? From the Sertoli cells, right? From the Sertoli cells. Do you think which are released? The sperms, the mature sperms, right? Sperms or spermatozoa, okay? They are released from the Sertoli cells, okay? They are released from the Sertoli cells. Do you think these are motile? Do you think these are motile? No, the sperms or the spermatozoa released from the Sertoli cells are not matured, or they are not motile. Okay, these are the sperms released from Sertoli cells are non-motile. They are non-motile. They are non-motile, right? They have tail, they have the acrosomal cap, they have the neck piece, but they are non-motile. Okay, so how they are released? The Sertoli cells, okay, along with the sperms, they will release a fluid called as testicular fluid. Okay, So this testicular fluid plus sperms will reach where? Will reach the they will reach the epididymis, right? They will reach the epididymis. When we discuss the epididymis, I told you maturation of the sperms occurs in epididymis, right? So in epididymis, the maturation of the sperms occurs, or the sperms will become motile. Okay, the sperms will become motile at epididymis. At epididymis, the sperms will become motile. Okay, they will get matured. Okay, so the sperms or spermatozoa which is released from the seminiferous tubules are what? They are non-motile. They can't move itself, right? Even though they have tail, they have energy, right? Mitochondria, they have head, they have acrosome, they have everything, but they are non-motile. Okay, so how the Sertoli cells will release them? The Sertoli cells will release a fluid called as testicular fluid along with the sperms, which will take them to where? Which will take them to the epididymis. Okay, from the epididymis, the sperm maturation occurs, or the sperm maturation occurs in the epididymis, where they will attain motility. Okay, epididymis, I told you it is a temporary storage, right? How long the sperms will get stored in the epididymis? Around 1 month. During that time, the sperm will attain maturity, right? It will attain motility. Okay, motility, and they are transferred to the ampulla of the vas deferens, where the sperms are stored. Okay, stored before ejaculation. Okay, stored in the ampulla of the vas deferens, where they are stored before ejaculation. Understand? So we will see the different organs in the sperms. Okay, I told you it has what? It has an acrosomal cap. Okay, this is normal cell only. Okay, it is like our normal cell only. So it will, what will it will have? It will have a plasma membrane. Okay, it will have a plasma membrane outside. Okay, it will have a plasma membrane outside, right? It will have a acrosomal cap, right? It will have a acrosomal cap. Okay, from where this acrosomal cap is formed? The acrosomal cap is formed from Golgi apparatus. Okay, it is formed from the Golgi apparatus. And what this acrosomal cap has? The acrosomal cap will have what? They will have the okay, we'll see the structure of sperm, right? We'll see the structure of sperm. I told you it is like a normal cell only, right? It is like a normal cell only. So since it is like a normal cell, it will be covered with what? It will be covered with a plasma membrane. So it has an outer covering of a plasma membrane, right? It will have a plasma membrane, right? It will have a plasma membrane. Then I told you it has an acrosomal cap, right? It has an acrosomal cap, right? Acrosomal cap, right? From where this acrosomal cap is formed? The acrosomal cap is formed from what? It is from the Golgi apparatus. Okay, from the Golgi apparatus. Then I told you it has a condensed nuclei, right? It has a condensed nuclei, right? What is the chromosome number in that nuclei? It is haploid, right? Or 23 chromosomes. Okay, right? 23 chromosomes, right? Then I told you there is a proximal centriole, right? Proximal centriole or the centriole, which will extend as what? Extend as the axoneme, okay, or axonal apparatus. Okay, in either side of the axoneme we have what? We have the mitochondria, right? We have the mitochondria. Okay, we have the mitochondria. Okay, on either side of the axoneme we have the mitochondria, or around the axoneme we have the mitochondria. Okay, then we have the elongation of this centriole, or elongation of the microtubule of the centriole, right? This is the microtubule. tubule of the centriole, right? And what is the structure? It is the tail of the sperm or flagellum of the sperm, right? Tail of the sperm, which is formed from the elongation of the microtubule of the centriole, right? It is the elongation of the microtubule of the centriole, right? So this is the acrosomal cap. What is this acrosomal cap contains? The acrosomal cap contains what? It contains the lytic enzymes, right? It contains the lytic enzyme. What is the function of this lytic enzyme? This lytic enzyme will help the sperm to penetrate the outer wall of the ovum, right? During the time of fertilization, this lytic enzymes will help the sperm to penetrate the outer wall of the ovum. Understand? The lytic enzyme will help the sperm to penetrate the ovum. Okay, and it is made up of Golgi apparatus. Acrosomal cap is made up of Golgi apparatus, which contains the lytic enzyme. Right. Now we has the nucleus, which I told you, it has a condensed nucleus with 23 chromosomes. Right. This part, okay, after the acrosome, this part forms the head. Okay, this part forms the head where the nucleus is present. Right. Where the mitochondria is present, it forms the neck. Right. And what is the function of mitochondria? This mitochondria will provide energy for the movement of the sperm. Right. Mitochondria will provide energy. Okay, it will provide energy. Mitochondria will provide energy. Right. It will provide energy for the movement of the sperm. Right. And what is the function of the tail? The tail will helps in the propel the sperms forwards. Okay, tail will propel the or moves the sperm further or move the sperm forwards. Okay, so the movement of the tail will propel the sperm forwards. Okay, so this is the organ which helps in movement, that is the tail. It will propel the sperm forward. Okay, understand each function of each and every organ of the sperm, right? This is the nucleus, that is which is going to fuse with the nucleus of the ovum to produce the embryo or zygote, right? The fertilization occurs with the fusion of the male nucleus with the female nucleus, right? And we have the acrosomal cap, which has the lytic enzyme, which will help the sperm to penetrate the ovum. Then we have the mitochondria, which will provide energy for the movement of the sperm, and we have the tail, which will propel the sperm forwards. Understand the function? Okay. Now we are going to discuss an important topic, that is regulation of spermatogenesis, right? Regulation of spermatogenesis. How the spermatogenesis is regulated? I told you, when is the time the spermatogenesis starts? The spermatogenesis starts at puberty. Spermatogenesis starts at puberty. Okay, starts at puberty. How? What happens in puberty? What happens in puberty? In puberty, there will be increase in what? Increase in gonadotropin releasing hormone, or GnRH. What is called as GnRH? It is the gonadotropin releasing hormone. That is the GnRH. Okay, gonadotropin releasing hormone. There will be increase in the amount of GnRH. Okay, increase in amount of GnRH from where? From the hypothalamus. Okay, from the hypothalamus there is increase in gonadotropin releasing hormone, amount of gonadotropin releasing hormone from the hypothalamus during puberty. So what happens? What happens is that this gonadotropin will go to where? It will go to the anterior pituitary, right? It will go to the anterior pituitary, right? Anterior pituitary, and causes the release of what? This will causes the release of two hormones, LH and FSH, right? Which is LH? It is the luteinizing hormone. Luteinizing hormone, which is FSH. It is the follicular stimulating hormone. So during the time of puberty, there will be increase in the amount of GnRH, which will give the signal to the anterior pituitary, or which will pass to the anterior pituitary and causes the release of what? Causes the release of LH and FSH. Okay, LS and FSH. Where this LH is going to act? LL. Okay, LH will going to act at the Leydig cells. Right, Leydig cells or interstitial cells, we have seen, right? Leydig cells, right? Leydig cell. And what the Leydig cells will produce? They will produce what? They will produce testosterone, right? It will produce testosterone, right? It will produce the testosterone hormone. Okay, this will produce the testosterone hormone. What's the function of testosterone? Testosterone is responsible for what? It is responsible for the spermatogenesis as well as the development of male sex organs or male reproductive system, right? It is responsible for spermatogenesis as well as the development of male reproductive system. Okay, so the LH will go and act at the Leydig cells, which will in turn produce testosterone, right? Which will helps in the spermatogenesis as well as the development of the male reproductive system. Okay, now we'll see where the SHS will go and act. SHS will go and act at the Sertoli cells, right? Sertoli cells. It will go and act the Sertoli cells. So what the Sertoli cells will do? It will produce a protein or a factor called as androgen binding protein. Androgen binding protein. What is this? Androgen binding protein. Okay, so the Sertoli cells will produce what? It will produce androgen binding protein. What is the function of this androgen binding protein? This androgen binding protein, they will increase the concentration of testosterone in the Sertoli cells or in the seminiferous tubules. This androgen binding Sertoli cells are where it is present in the seminiferous tubules, right? Where the spermatogenesis occur? It is occurs in the seminiferous tubules. What is need for the spermatogenesis to occur? We need testosterone. So this androgen binding protein will helps in concentrating testosterone inside the seminiferous tubules. Okay, they will helps. So these two will help in what? Will help in spermatogenesis. Right? How? The androgen will helps in concentrating. It will help in concentrating the testosterone inside the seminiferous tubules. Right? So this testosterone will in turn helps in spermatogenesis. Okay? Spermatogenesis. Understand? Right? So if this is occurring like this way, okay, what happens if we have sufficient sperms? Or if we have sufficient testosterone to produce sperms, what happens? When the amount of testosterone becomes sufficient, okay, when the amount of testosterone becomes sufficient, what happens? A negative feedback, right? A negative feedback or a negative signal to stop the testosterone production will be sent to where? Will be sent to the pituitary as well as hypothalamus. Okay, when the amount of testosterone becomes sufficient to form spermatogenesis as well as for the development of the reproductive organ, what happens? The testosterone, when the body will send signal, right, a negative signal to the hypothalamus and the pituitary to stop the production of their respective hormones. Okay, it will send a negative signal. Okay, it will send a negative, or this is called as what? This is called as feedback inhibition. Right, it will inhibit. A feedback is sent, right? We have enough testosterone. So a feedback is sent, we have enough testosterone. What this feedback will do? It will inhibit the production of the particular hormones. Okay, it will inhibit the production of the particular hormones. Okay, so a negative feedback is sent, right? So that there will be decrease in GnRH, decrease in LH, right? Decrease in testosterone, right? Decrease in testosterone, thus decrease in spermatogenesis. Okay, what about this one? What about here? The Sertoli cells, or what? If we have enough number of sperms, or the spermatogenesis is enough, what will happen? The Sertoli cells will produce one factor called as inhibin. It will produce a hormone called as inhibin, right? I told it will produce a hormone called as inhibin. This inhibin will go and inhibit the hypothalamus as well as the pituitary. This will go and inhibit the hypothalamus and pituitary to decrease their secretions. Okay, to decrease their secretion. So the feedback is going from the testosterone, right? When the level of testosterone is sufficient, if negative feedback will go. Similarly, when the production of sperms are sufficient, a negative sperms are sufficient, inhibin is produced to inhibit the further spermatogenesis. Okay, what is the function of inhibin? To suppress the spermatogenesis, right? The function of inhibin is to suppress the spermatogenesis. So when the spermatogenesis becomes sufficient, what happens? Inhibin is produced, which will inhibit, will send signal to the hypothalamus and pituitary to decrease their secretions. Okay, so when these two negative feedbacks are sent, what happens? There will be decrease in the GnRH, or gonadotropin releasing hormone, in the hypothalamus, right? So there will be decrease in the FSH and LH secretion from the pituitary. There will be decrease in testosterone. There will be decrease in FSH. So there will be decrease in the androgen binding protein, right? So in turn there will be decrease in the spermatogenesis. Understand? This is how spermatogenesis is regulated, right? So if there is increase in GnRH, what? It will increases the spermatogenesis, right? So when sufficient spermatogenesis has occurred, or sufficient testosterone is formed, what? There will be a negative feedback, right? Negative feedback, and is negative feedback is sent to suppress the GnRH and LH and FSH secretion, so that the production of sperms are reduced, or spermatogenesis is suppressed. Understand how this is regulated? Okay. Right, this is how the spermatogenesis is regulated. Okay, so the sperms plus the secretions from the accessory ducts. Okay, secretions from where? Secretion from the accessory ducts plus where? Plus the accessory glands. Accessory glands will together gives what? Will together gives the semen, right? The secretions of accessory glands and accessory ducts, we have seen that they are needed for the proper maturation, activation and the movement of sperm, right? It is needed for the perfect maturation, activation and the movement of the sperm, right? So this sperms plus the secretion from the accessory ducts and glands together constitute what? Together constitute the semen. Okay, so what is the sperm count per ejaculation? It is around 200 to 300 million sperms, okay, per ejaculation. Okay, million sperms are released per ejaculation. Okay, out of this 60% of the sperms, okay, out of this the 60% of the sperms should have the normal size and shape. Okay, size and shape. Okay, 60% of the sperms should have the normal size and shape, and 40% should show the vigorous motility. Right, the 60% sperms should have the normal size and shape, and 40% sperms should show the vigorous motility for fertilization to occur. Okay, to fertilization to occur. Okay, so the per ejaculation 200 to 300 million sperms are released. Okay, out of which 60% of the sperm should have normal, it should have normal size and shape, and 40% should show vigorous motility, so that the fertilization should occur. Okay, this is called as this is what is the spermatogenesis. Understand the process of spermatogenesis, right? But from where the spermatogenesis are formed, which is the first cell? It is the primordial germ cell, present in where? Present in the embryonic stage, right? It will migrate to the respective gonads, right? So in spermatogenesis, the primordial germ cell will reaches the testis, right? primordial germ cell will reaches the testes. Then we have seen at the time of puberty, the male germ cell which is present in the seminiferous tubules will start at undergoing mitotic division, right? It will undergo the mitotic division and forms what? Forms the primary spermatocyte. This primary spermatocyte will undergo first meiotic division to form secondary spermatocyte. This will undergo the second meiotic division to form spermatids, right? The process of conversion of the spermatid to spermatozoa or sperms is called as what? It is called as spermiogenesis, right? And the release of the sperms from the Sertoli cells is called as, spermiation, right? Then we have seen the regulation, how LH, FSH and testosterone and inhibin, okay? What are the 4 hormones? No, what are the 5 hormones? We have GnRH or gonadotropin releasing hormone from the hypothalamus. We have the LH and FSH from the pituitary. We have the testosterone from the Leydig cells. We have the androgen binding protein and inhibin from the Sertoli cells. Okay, these hormones together regulate the process of spermatogenesis. Okay, clear? This is the process of spermatogenesis. Clear, everyone?